Now let us start our study with the growth promoters. As described earlier, growth promoters are the chemical substances which help in initiating, stimulating the growth development within the plant. Thus, there are mainly three types of growth promoters. First, auxins. Second, gibberellins. Third, cytokinin. So let us begin our study with the first, auxins. Auxins are the growth promoters which can be either natural or synthetic. They were first isolated from the human urine. It is such a chemical which helps in the promotion of the growth within the plant. Moreover, in plants, it was first shown it in effect in oat coleoptile. Thus, within plants, oat coleoptile was the first in which the effect of auxins was shown. As said, there are mainly two types of auxins. First, naturally synthesized, that is natural auxins, and second, synthetic auxins, which are artificially synthesized. The best examples of natural auxins include IAA, that is indole acetic acid, and synthetic auxins is NAA, that is naphthalene acetic acid. The other examples of these auxins include IBA, that is indole butyric acid, which is an example of natural auxin, and the other example of synthetic auxin includes 2,4-D, that is 2,4-dichlorophenol acetic acid, which is also known as dichlorophenoxyacetic acid, which is an example of synthetic auxin. Now let us study about the various effects of the auxin. There are many positive effects of the auxin. For example, it helps in stimulation of the flowering process, that is stimulates the process of flowering. Moreover, it also stimulates the respiration process. The third effect includes that it stops, that is, it helps in stopping the premature fall of fruits and leaves. It also acts as weed controller, that is acts as weedicide, and thereby it helps in eradication of weed and so also act as weed eradicator. Moreover, it is also useful in stimulating the cell division and tissue culture. Apart from that, it helps in regulating the phototropic movements, that is, helps in performing the phototropic movements of the plant organ. And thereby helps in its movement. It also helps in the formation of adventitious roots, apical dormancy, and helps in the development of seedless fruit. And this development of seedless fruit is also known as parthenocarpy, which we have already studied earlier in the previous unit. Thus, these all influence the effects of auxin, that is a growth promoter within the growth of the plant. Now let us start our study with the second type of growth promoter, that is gibberellin. This is a growth promoter which helps in the stimulation of the growth of the plant. Such a promoter was first identified in Japan in a paddy diseased plant, which was known as bakane, that means a foolish plant. This figure depicts the bakane or paddy diseased plant. It is caused mainly by an organism known as Gibberella fungus, which secretes a substance known as gibberellin. And from there it was first identified as a growth promoter. As it can be seen from the figure, such a diseased plant is normally long, narrow, thin, yellow and sterile. After this, the gibberellin was also identified in other plants, which is caused by various fungus and other higher plant secretion. Thus, it was identified in many other plants later on, which was secreted by about more than 100 types of fungi and other higher plants. Like other growth promoters, gibberellins are also of different kinds and they generally occur in form of GA1, GA2, GA3 and however it goes on in such a manner continuously. Such gibberellins, that is growth promoters, are generally acidic in nature and their synthetic action increases in darkness. The activity and effect of such an gibberellin, that is growth promoter, is entirely different from that of auxin. Now let us study the effects of this growth promoter. First, it induces flowering in some plants. Since it is a growth promoter, it will definitely enhance in many manners in order to induce the growth of the plant. Apart from that, it also helps in removing the expression of the genetic dwarfism. That is the dwarfism character that is exhibited due to genetic disorder is also removed with the help of this growth promoter. Moreover, it is also responsible for removal of dormancy of various buds and seeds. It also helps in stimulating the synthesis of various enzymes which activates the embryo. Moreover, it also helps in increasing the leaf area. Thus, these are the effects that is exhibited by induced secretion of the gibberellin. Now let us start with our third growth promoter, that is cytokinin. Such a growth promoter is generally present in the region which is highly involved in cell division. The cytokinin helps in the division of cell process. Cytokinin was first identified as a substance kinetin, which is a form of adenine that was first obtained from the sperms of herring fish. This cytokinin was first originally obtained in the form of kinetin. After that, a substance called zeatin, which was naturally present within maize and coconut milk, possessed features and properties same as that of the cytokinin. And because of this, it was possible to naturally extract the cytokinin and its effect. However, before that, it was not possible to obtain cytokinin in a natural manner, and so synthetic cytokinin were prepared. But after the discovery of zeatin, it was possible to obtain cytokinin in a natural manner. Now let us study the effects of cytokinin. As said earlier, it is generally present in the region of cell division and so it helps in the growth of the plant. Thus, it stimulates the process of cell division, elongation, and differentiation. It also helps in reducing the apical dominance nature of the plant. And also helps in reducing the senescence of the plant. That is a property which generally leads to the death of the plant, which we'll study later. Moreover, it also retains the chlorophyll in leaves, which can be said as its disadvantage. Apart from that, it also helps in the translocation of nutrients and other organic substances. So this was all about the growth promoters. Now let us study about the growth inhibitors.